Hello, everyone, and welcome to today's live broadcast, investigating DNA repair mechanisms in hypoxic brain tumors and the link to treatment resistance, presented by Sophie Cowman, fourth-year PhD student, Institute of Integrative Biology, University of Liverpool. I'm Alexis Corrales of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Sophie Cowman. Sophie is passionate about understanding how the tumor microenvironment can influence DNA repair, which is the focus of her current research. In 2011, Sophie commenced a BSc in molecular biology at the University of Liverpool. In 2012, she was awarded a competitive scholarship enabling her to undertake a summer research internship in the lab of Dr. Geraint Parry, investigating the nuclear pore complex. Throughout her PhD, Sophie's research primarily utilizes cell culture techniques, gene expression analysis, and advanced confocal microscopy to investigating DNA repair mechanism in hypoxic brain tumors. For a complete biography on our speaker, please click on the Speaker Biography tab. Welcome, Sophie. You may now begin your presentation.
Thank you for that introduction. So hello, my name's Sophie, and I'm a final year PhD student in Violaine group at the University of Liverpool. And I'm here to talk to you today about my work on the impact of hypoxia on DNA repair mechanisms in brain tumors. So our lab group primarily focuses on looking at the impact of hypoxia on biochemical pathways within cancer cells. And if you'd like to connect with me on Twitter, here's my Twitter handle. Please feel free to follow me if you'd like. So what is hypoxia? So the classical definition of hypoxia is that it's the point at which the demand for oxygen outweighs the supply. So there's basically a deficit in the oxygen supply to the environment. And there's many examples of hypoxia, such as when you start to walk up a mountain and you get lower air pressure, there's less oxygen available for you to breathe. So then you start feeling unwell, and this is altitude sickness, and this is because you are becoming hypoxic. Also, on a more tissue-specific level, in a heart attack, a blood vessel becomes blocked, and this prevents blood flow to a region of the heart, and the cells can start to die due to the fact that they have no oxygen available. And this is what actually causes people to die after a heart attack. And the final example I want to talk about is tumor hypoxia. So tumors in themselves can experience a hypoxic environment, and this can have a very big impact on how they respond to treatments. So tumor hypoxia is going to be the focus of this webinar. So when we think of the structure of a solid tumour, we have to understand that there's actually gradients throughout the tumour. So for example, there's a gradient from the outside to the centre of oxygen, with a high oxygen environment on the outside and a lower oxygen environment on the inside. But also there's pH gradients and nutrient gradients. And so that means that the cells in the centre are having a very different environmental experience than the cells on the outside. And this can result in the formation of a necrotic core, where cells are more likely to die in the centre due to the lack of oxygen and nutrients, for example. And so our group primarily focuses on this oxygen gradient, where we have high oxygen in some areas and low oxygen in other areas. And so we actually focus on hypoxic tumours. So hypoxic tumour examples include breast cancers, liver cancers, and brain tumours, for example. And it tends to be the more solid tumours that are hypoxic, so the large, solid tumours. So actual oxygenation of tumours and general tissues is very dependent on where it is in the body. So most of you can appreciate the fact that in your lungs, the oxygen is probably going to be higher than other areas of your body because you're actively breathing oxygen. So we work on brain tumours, And in the brain, in the normal environment, so Hypoxia, so that means basically the normal healthy tissue, there's a median oxygen concentration of 4.6%. And this can actually go up to about 8%, depending on whereabouts in the brain you're taking the measurements from. However, in brain tumours, the median oxygen percentage is actually only 1.7%, so this is quite low, meaning that they're actually hypoxic compared to the healthy tissues. So there's a number of causes of tumour hypoxia. So when we think of a solid tumour, we have to understand that these cells are rapidly dividing to form this mass. So they're dividing a lot quicker than normal cells to create the solid lump, basically. And so this results in these long diffusion distances between cells and vessels because the vessels can't grow fast enough compared to the cells. So this means that oxygen in the vessels can't diffuse to the cells, making them hypoxic. Also, the blood vessels that do form tend to be quite weak. So this means that they're more prone to bursting and also becoming blocked, which may result in a specific area of the tumour having a reduced blood flow and a reduced oxygen concentration and oxygen environment. So throughout the tumour, there's different pockets of different oxygen concentrations, and this is a very transient process. So for example, at one point an area could be quite well oxygenated, but due to a blocked blood vessel, this can actually dramatically reduce. So there's a number of consequences of tumour hypoxia, particularly from a cancer perspective, and they're very, very important consequences when it comes to clinical treatment. So the first consequence I'd like to mention is this idea that hypoxic cells are more likely to invade tissues and form metastasis. So they can travel from the primary tumour site, invade through local regions of tissue, find blood vessels, enter them, and travel to a secondary area and form metastasis. And this is a very big problem in the clinic because metastasis is often the thing that kills people. Additionally, a second consequence of tumour hypoxia is resistance to radiotherapy and chemotherapy. So this means that the cells that are being hit with these anti-cancer treatments, but they're going on to live on and healthily divide as this diagram depicts. So actually they can end up forming, and further enlarging the tumour as such. So overall, hypoxia has a really big impact on so many different biochemical pathways, and this really impacts how the cells behave and the phenotype of the cells. And when we think of the impact of hypoxia on biochemical pathways, we have to consider the hypoxia-inducible factor, which is known as HIF. So HIF is a transcription factor which is primarily responsible for the adaptation to this low cellular environment. So under what we call normoxia, which is basically a normal oxygen environment for that cell or tissue, we have these enzymes called PHCs, or prolyl hydroxylases, which use molecular oxygen to actually hydroxylate HIF, which then targets HIF for destruction. So HIF isn't able to activate hypoxia-inducible genes. However, under hypoxic conditions, PHCs don't have this molecular oxygen available, so then HIF isn't targeted for destruction by hydrolysis, and it can actually go on to activate a number of different genes involved in different biochemical processes. So, for example, a few processes that HIF has the ability to induce is, for example, erythropoiesis and angiogenesis, and these two processes are very key for making more blood vessels and making more blood cells to try and reoxygenate the cell, and also these metabolic changes such as switch to glycolysis. So HIF can actually affect a number of biochemical pathways within the cell, and we don't yet know how many targets HIF actually has. And there's a lot of work going on at the minute to kind of understand what is targeted by HIF and how it is influencing the expression of genes and the consequences of that from a cancer perspective. So I'm now going to move on to kind of talk about the brain tumour models that we use. So brain tumours are very problematic, so they're killing many people, and it's really important that we kind of get a better understanding of brain tumour cell biology so we can improve treatment methods. So the first tumour that I work on is adult glioblastoma. So this is the most common primary brain tumour in adults, and the survival rate is very poor. So as you can see by the 5-year survival rate, it's so poor, and most people actually only survive 2 years after diagnosis. Whereas pediatric medulloblastoma, the survival rate is much better, but we have a lot of problems because when we start to use chemotherapy and radiotherapy on a child's developing brain, we start to get issues such as hearing loss and speech problems and learning difficulties. So overall, it's really important that we kind of get a better understanding of these tumours and what's going on with the cells. And both of these tumours are very, very hypoxic. So when it comes to treating cancers and treating things like brain tumours, we kind of have a few treatment options. So in general, for many types of tumours, we kind of follow this pathway, so which involves basically surgically removing the tumour mass and then followed by chemotherapy and radiotherapy. And the order of this sort of treatment can vary and often depends on the circumstance and the cancer type. So for glioblastoma, known as GBM, we initially try to remove the tumour as much as possible, and then we treat with a drug called temozolomide. However, the temozolomide is not very effective, and it is actually only successful against certain tumours which have this silent MGMT enzyme. And this is probably a good explanation as to why we have such a poor survival rate for this tumour, because we have very limited treatment options. Additionally, we use hyperfractionated radiotherapy in conjunction with the temozolomide treatment. So the way hyperfractionated radiotherapy works is that instead of giving a very large dose of radiotherapy on one day and waiting, for example, for a month before you give another large dose, they actually split that large dose up into small amounts and give it regularly, for example, daily, over a number of weeks. And this has proved a lot more successful than just, irradiating on one day, as it really helps to try and remove the cancer cells and kill the cancer cells, and also seems to be less problematic with regards to side effects. So for medulloblastoma, so MB, medulloblastoma, we also try to remove the tumours. And we've got a lot more drug-related options to treat this tumour, which is probably why we have a better survival rate compared to glioblastoma. So also for medulloblastoma, we additionally use hyperfractionated radiotherapy. And there's kind of been some recent discussions, particularly in the media, about using this proton beam therapy. So the potential benefits of proton beam therapy is that the irradiated volume is much more specific. So this means that we have a lot less damage to healthy tissues in the brain. So this could potentially help reduce with all those side effects and those problems such as hearing loss and speech defects. However, there's a lot of research going on in the minute to try and understand whether proton beam therapy actually helps any more than, the standard traditional radiotherapy, because we don't know whether it's actually more effective at killing the cancer cells. So as I mentioned previously, a big consequence of hypoxia and tumour hypoxia is this resistance to treatment. And so what we wanted to do is we wanted to take our brain tumour cell line, which we work on, and try and understand whether we can see this resistance phenotype induced by hypoxia. So what we did is we took brain tumour cell line, so medulloblastoma cell line, incubated it in 1%, which is hypoxia, or 21%, which we define as normoxia, or its atmospheric oxygen environment, for either a day or for 5 days. Then we treated the cells with either a drug or a radiation. And then finally, we assayed cells for viability after the treatment duration. And this is an example of the data that we obtained. So this is with the etoposide treatment. So the dark orange bar is normoxia. And as you can see, with the etoposide treatment, it's around about 30% viability after the treatment. And with 1 day hypoxia, there's a similar sort of level of viability. However, with 5 days hypoxia, so which is a much more chronic hypoxic environment, you can see the viability is a lot higher, looking at like 70 to 80%. So this is implying that this hypoxic environment for 5 days has made the cells more resistant to treatment. And what we got from this is the fact that it requires chronic hypoxic exposure, so a long-term 5-day exposure to cause this resistance, yet acute hypoxia wasn't able to do this. So when we think of cancer treatment, we have to consider DNA damage and DNA repair, because many treatments actually aim to damage DNA repair, damage the DNA itself and cause DNA repair pathways to be triggered. So here we have the DNA, which is attacked by a drug or a radiation, and this can create different types of damage, including break, oxidation, hydrolysis, and cross-linking. And it's very important to understand what type of damage is being induced by a drug or a radiation, because these all activate different repair pathways. And overall, when we're considering repair, we have to understand that DNA repair mechanisms are really influenced by genetic background and the cellular environment. And kind of these ideas led to our research question. So what we want to understand is, how is hypoxia influencing these DNA repair mechanisms in brain tumours? Because DNA repair is highly influenced by cellular environment, so is a low oxygen cellular environment impacting the mechanisms? And this is what we'd like to understand. So I'm going to kind of present you kind of two parts of the story. One's a very, very specific story looking at double-strand break recognition in medulloblastoma, whereas the second part is very much ongoing work looking at multiple repair pathways which are downregulated by hypoxia. So moving on to part one. So what we decided to do is we'd seen this treatment resistance, this phenotype of resistance in hypoxia, and we wanted to know what was causing that. So we decided to do a microarray experiment with cells, so this is with medulloblastoma cells. We put them in hypoxia over varying time points, and what we found is that many genes were actually regulated at later time points. And in the literature, it's often described that hypoxia has the ability to induce the expression of multi-drug resistance genes, so MDRs. So what we thought was, okay, so maybe this resistance phenotype is actually due to the induction of multi-drug resistance genes. So we took our microarray data and pulled out the expression levels of 3 key multi-drug resistance genes. However, what we found is that none of these genes were induced by hypoxia. Therefore, this resistance phenotype was not related to multi-drug resistance. So this made us think, okay, so as treatment causes DNA damage, maybe something's going on with the DNA repair mechanisms. So we specifically took the expression profiles for a number of DNA repair pathways. So for example, the non-homologous end joining pathway and homologous recombination repair. So both of these repair pathways involved in the repair of double-strand breaks. So the non-homologous end joining is primarily in G0 and G1, whereas the homologous recombination is in S phase in G2, when there's a homologous DNA strand available. So we looked at the expression and we found that a lot of the genes were strongly regulated at later time points in hypoxia, so more chronic environments. And in particular, we found that there was a clear downregulation in key genes involved in double-strand break recognition. So I'm going to kind of start to explain of how double-strand breaks are recognized in the homologous recombination pathway. So here we have the DNA double helix, which is attacked by a drug or a radiation, and a double-strand break forms. And this basically means that both strands of the DNA have completely severed. So this is the most lethal type of DNA damage to a cell, and one double-strand break can actually kill a cell. So the initial step of double-strand break repair is recognition, and this is done by the MRN complex, which is basically recruited to the site of damage. So the MRN complex is made up of nibrin, or NBN, MRE11, and RAD50. And the role of these proteins is to initially sense the damage. They are DNA damage sensors. So next in the pathway is recruitment of signal transducers, and in particular the signal transducer ATM, which is recruited to the site of damage and phosphorylated, which activates its kinase abilities. So ATM is then able to either phosphorylate targets which lead to repair, such as CHEK2, and then the DNA damage will be repaired by the homologous recombination pathway, or alternatively, it can force the cells into an apoptosis pathway via P53. So in summary, we have the double-strand break formation, we have the break recognition, we have the signal transduction via ATM, and then either we have repair or apoptosis. So as I mentioned previously, we have this downregulation in some of the key components of the MRN complex responsible for recognition of breaks. So we took our microarray data and found this, and then we decided to look using real-time PCR, and clearly here you can see this nibrin and this MRE11 are downregulated by hypoxia, whereas this RAS50 isn't. And we wanted to see whether this was purely due to the fact that it was at later time points, or maybe earlier time points would trigger this. However, you know, at 6 hours, you don't really see this downregulation until you get to the later time points, like 48 hours plus. So it is more of a chronic thing, more of a chronic hypoxic exposure that is causing this downregulation. And as this was all at the gene expression level, and obviously repair is always with proteins, we needed to check the protein levels of these key DNA repair proteins. So we did Western blots, and as you can see here, particularly with the quantification, there's clear downregulation of both nibrin and MRE11 at the protein level, and it's mirroring the gene expression data. So what we have is this downregulation of this double-strand break recognition complex, which is kind of the very key initial steps in the repair pathway. So we wanted to see whether ATM was affected, so the next steps in the pathway, and whether the activation of the ATM via this phosphorylation is impacted by hypoxia. And indeed it was. So as you can see by this Western blot, so in the 21%, we've got a nice amount of ATM and a nice amount of phospho-ATM. And then at 1%, it starts to decrease. And when you go to even more severe hypoxic environment, it's even more decreased than that. And you can see this by the quantification. So clearly, particularly in the total ATM levels, you can see this nice downregulation, which increases with the severity of hypoxia. So this kind of made us think, if ATM is also affected, maybe P53 is affected, and maybe any effect on P53 is really impacting how the cells are responding and maybe contributing to resistance. So we looked at the induction of the targets of P53. So looking at this Puma, MDM2, and P21. So this is the mRNA level. And what we found in hypoxia, there was less activation of P53 targets. And we think this is primarily due to the fact that in hypoxia, there's less phospho-P53, so that means that there's less stable P53 available. So in summary, what does this all mean? So under hypoxic conditions in our medulloblastoma cell line, we have this downregulation of the double-strand break recognition complex, which also leads on to the ATM being downregulated and a reduced activation of ATM. And we don't have stable P53 and activation of P53 targets. So what we think is happening is that these cells just aren't sensing the damage and recognizing that it's present. Therefore, they're not triggering apoptosis, and so the cells are still surviving. And this is what we think is explaining the treatment resistance phenotype that we observed. However, this mechanism, although it's very good at explaining how this treatment resistance may occur, this wasn't very universal, and it wasn't in all our cell lines, and it wasn't in all our cell types. And in particular, we wanted to kind of further investigate the effects of hypoxia on glioblastoma. So we decided to kind of take a step back and have a more global view of what's happening to the DNA repair genes under hypoxic environment. So this leads on to part 2, where I'm going to talk about data from multiple repair pathways, but I'm going to focus in on the error-prone double-strand break repair pathway, so non-homologous end joining. So what we decided to do was we decided to incubate a number of glioblastoma cells in different oxygen conditions. So normoxia, which is 21%, 1% which you could define as moderate hypoxia, and then more severe hypoxia, which is 0.1%. And then we extracted RNA from these cells and we subjected them to a nanostring experiment. So a nanostring experiment basically enables you to look at the gene expression of a number of genes in a single reaction. So we decided to look at 180 DNA repair genes in many different types of DNA repair pathways. And today I'm going to focus on non-homologous end joining. So how does non-homologous end joining work? So we have a double-strand break. And as I said before, non-homologous end joining is only in G0 and G1, so it doesn't have the requirement of a homologous strand to be present. So we have the double-strand break. The ends are processed through the recruitment of two kind of key protein complexes, so Q70 and Q80 initially, and then DNA PKS. And then this goes on to further end processing by XRCC4, and then ligation of the strands back together by ligase 4. So this is quite a simple DNA repair pathway. It basically just sticks the ends back together and can actually cause quite a lot of errors in the sequence. So when we actually look at the data for the expression of these genes, what we find is that ligase 4 is strongly downregulated by hypoxia. And this downregulation actually increases with increasing severity of hypoxia. And we found this in nearly all of our glioblastoma cell lines. So when we look back at the pathway, here we have this ligase 4, which is downregulated, shown by the red arrow. And therefore, we think that maybe the strands of DNA aren't actually being joined back together as they should. And we wanted to kind of look at the consequences of this and what was going to be happening in this situation and what would this mean for the cells. So we decided to take a live cell imaging approach to look at repair protein recruitment. So what this involves is basically we tag DNA repair proteins with a GFP molecule, so we can visualize this on a microscope, transfect it into cells, and then we irradiate the cells with a multiphoton laser. And the multiphoton laser has the capability of inducing double-strand breaks to the DNA in a very specific defined region. So here we have, for example, the nucleus, which is green due to the fact that we've got a GFP DNA repair protein. And then we choose a specific region of interest to blast with the laser. And then what we should hopefully see is basically an increase in intensity of the GFP signal at the site of damage as the protein is actually recruiting to the double-strand breaks and trying to repair them. So you'll see this increased intensity. So you're now going to see a video kind of explaining this, and hopefully you'll be able to really visualize this increase in intensity over time. What we can basically do is we can actually measure this. So we can measure the kinetics of the initial increase in the protein signal, so in the GFP signal, and it appears that between hypoxia and normoxia, the actual initial recruitment of protein is very, very similar. However, at later time points, you can see this deviation between the two lines, where hypoxia seems to the protein seems to be remaining there and not being lost, so it's not dissociating from the breaks. And this could suggest that the repair is less efficient in hypoxia. And this work is very, very much ongoing, and that in the coming months we hope to kind of understand this a little bit further and kind of take this on to later time courses to really understand whether that protein isn't dissociating from the damaged site. So what we found is we found a number of other changes in hypoxia to multiple DNA repair pathways. So I've just shown you non-homologous end joining as an example. However, we found changes in mismatch repair, base excision repair, homologous recombination repair, for example. And we want to kind of understand what is the functional impact of these changes. What does this mean for DNA repair, and what does this mean for the cells? So to do this, we kind of use multiple methods. So, for example, classical biochemistry techniques such as Western blotting, qPCR. Then we've also employed our microscopy technique using this laser for damage. And finally, we've started to use a thing called the Comet assay, which is a very traditional assay used in DNA repair studies. So I'm going to talk to you briefly about what the Comet assay is and how it works and how it can be very, very useful to look at DNA repair kinetics. So how the Comet assay works is you take some cells, you treat them with either drugs or irradiation, and then you mix those cells with low melting point agarose and put that blob of cells and agarose onto a microscope slide. Then you apply electric current through the agarose, very similar to how you would with PCR and gel electrophoresis, and then you stain the DNA and you can visualize it on a microscope. So, for example, here in the untreated, you get this really nice tight circle of DNA, whereas in the treated, you get what looks like a comet, which gives the assay its name. So you get a nice little tight circle at one end, which is the head end. However, then you get this like balloon shape at the back. And this is due to the fact that the DNA has breaks. And because the electric current's been applied, it's allowed the DNA to migrate through the gel. And this kind of gives us an idea of the extent of damage within the cells. And so what we can do is we can use this to understand how long it takes to repair that damage. So, for example, you take some untreated cells and then some treated cells. So you can see untreated is very circular, whereas the treated are more like comet shapes. But you can allow the cells to repair after treatment. So, for example, 30 minutes after repair, you can see a few comets are still there, but they're a little bit smaller. Whereas at 60 minutes after repair, it's basically reverted back to the untreated. So this gives us a time frame of how long it takes for these cells to repair. And what we want to do is compare normoxia and hypoxia and see whether this time period changes. And that will give us a very good indication of how effective the DNA repair mechanisms are, especially when all these changes to the DNA repair genes and proteins have occurred in hypoxia. So to summarize this part, what we've seen is multiple DNA repair pathways are downregulated by hypoxia in glioblastoma cell lines. And we really want to kind of answer this question of what are the consequences of these changes. And to do this, we're using a number of methods, including the live cell imaging, looking at the DNA repair protein recruitment and dissociation, and also this Comet assay to look at the DNA repair kinetics. So this is very much work to be continued, and we hope to get some more answers in the next few months. So in summary, not all cells were responding to hypoxia in the same way when it comes to the changes to DNA repair mechanisms. And this is very important because from a clinical perspective, we really need to understand how different cell types are responding to hypoxia and how this might affect how they respond to treatment. So today I presented you two potential mechanisms which are induced by hypoxia. So the first, looking at the downregulation of double-strand break recognition, and the final one, look at a bit more broad, looking at multiple DNA repair pathways. So the next steps from this are really to kind of understand how are these changes in the DNA repair proteins regulated by hypoxia? What is directly, mechanistically, causing these changes? So in the literature, when you look at changes to DNA repair proteins by hypoxia, there's kind of three main causalities. So the first is this direct reduction of gene expression, so something's acting at the promoter to reduce the expression. Or alternatively, it could be epigenetic changes or even post-translational modifications of the DNA repair proteins. And kind of the overriding question is the hypoxia-inducible factor, HIF, involved in this story? And these are the sort of questions that we hope to answer in the next few months, because if we truly understand the mechanism behind these regulations, then we can actually start to manipulate the system and manipulate the pathway to try and kind of understand it further to try and improve treatment methods. So I really hope you've enjoyed gaining an insight into how we look at hypoxia and brain tumours and their DNA repair mechanisms. Thank you very much for listening. I'm more than happy to take any questions. Thank you.
Thank you, Sophie, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, why is acute hypoxia not sufficient to induce these changes?
So the answer to this isn't very simple, unfortunately. It's very dependent on which changes we're talking about. So some changes we do see in acute time points, whereas some are more at a later time point. And we think this is because that long-term hypoxia is inducing many genetic changes, such as epigenetic modifications, that require a time period for those to occur, which results in the changes to the DNA repair mechanisms that we see. So the answer isn't so clear-cut, but we have some ideas as to why that might happen.
Okay, Sophie, our next question is, how did you make all of these measurements? Under hypoxic conditions or after bringing the samples out of a 1% O2 environment into room air?
So we are actually fortunate enough to have what's called a hypoxic glove box. So this is an incubator where you can actually put your hands in it and culture cells within the actual chamber. So they are maintained within the hypoxic environment. So this means that a lot of the analysis that we have done, such as the testing of different drugs and things like that, has been done under the hypoxic environment. There is some experiments where there was a period of reoxygenation. However, we think this impact is very small because a lot of the changes that we see to DNA repair mechanisms are maintained for a period of time after reoxygenation.
So the second part to the question. So I'm assuming you did all these measurements, analysis, experiments in room air after bringing the samples out of the incubator. So wouldn't it be more accurate to describe these results as being potentially a result of reoxygenation as opposed to hypoxia alone?
So it's very important to consider the fact that sometimes we have reoxygenation. However, as I said before, a lot of the experiments are done under permanent hypoxic conditions. However, when we do have reoxygenation, we try and account for that by allowing the cells to equilibrate so that any oxidative damage is repaired, for example. But most of our experiments are done under a hypoxic environment.
Our next question, how are these changed DNA repair genes regulated in hypoxia?
So that's kind of the big question that we're trying to answer at the minute. There's many documented pathways that result in DNA repair gene regulation in hypoxia. However, it appears to be different for each different type of gene, each different pathway as well. So there's some suggestions that these are via HIF, a direct mechanism of HIF, for example, impacting the promoter of the genes. However, there's other mechanisms where, it's completely independent of HIF. And this is something that we really want to explore, because as soon as we understand how the genes are regulated, we can maybe start thinking about how to target these mechanisms for therapeutic benefit.
Do you think this could have an impact on the way we treat these tumors in the clinic?
That's the ultimate hope, because when we think of the clinic, we have to understand that all these drugs that they're using in the clinic for these tumors are actively damaging the DNA. And if those tumors are hypoxic, something is going to happen to those DNA repair mechanisms, which really might impact how those tumors are responding to the treatment. So we really do hope that we're going to, develop this further, and it'll kind of give us a better understanding of what's going on in these tumors, and then we can maybe translate this into the clinic in the future.
So for your next question, hypoxia can affect the cell cycle. Do you see this in tumor cells? Could this have an impact on your findings?
So in the literature, it's quite well documented that hypoxia does have an effect on the cell cycle. But what we found is that this is actually very cell type specific. So a study from our lab looked at glioblastoma cells. We took lots of cell lines as well as primary cells that have been taken from tumors. And we found that actually hypoxia did not have an impact on the cell cycle. And this is really important finding, because when you have impacts on the cell cycle, this can alter the response to drugs, because often the killing mechanism of drugs requires cycling cells. So our cells aren't actually impacted with regards to the cell cycle and hypoxia, which is a positive thing.
And Sophie, it looks like we have time for one more question. Some drugs don't work as well in hypoxia. So do you know if you are getting the same amount of damage in the hypoxic cells as the normoxic cells?
Yeah, that's a very important question. So what we do is we actually use the Comet assay, which I described in the talk, to measure the level of damage that we're achieving in the cells. And we found that with most of our drugs that we use, we do get the same amount of damage in hypoxic cells as normoxic cells. However, there's some drugs that really don't work in hypoxia, so we have to do these treatments out of the hypoxic chamber. And we have to consider, the impact of the reoxygenation within that experimental setup. So, yeah, we kind of do look into that, and it's a very important thing to consider.
Thank you again, Sophie. Do you have any final comments for our audience?
No, just to say thank you very much for listening. I hope everybody enjoyed the webinar.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of the registration. We would like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through October of 2018. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.